This is a production of Cornell University.
Thank you very much. It's thrilling to be back here. I have wonderful memories of my graduate days here. I have wonderful memories of life in Ithaca. My oldest daughter was born here, so that's always a special place to come back here. Tried to bring them here, but it's a little harder flying 4 people over the Atlantic than just one, so we decided not this time, but maybe another time. But I'm very honored to be invited back to give the seminar. Thank you so much, Teresa and Gillian, for the kind invitation. Thank you so much, Lou, for all the organization that goes alongside of this. And today I'm going to talk to you about my work on, like Teresa said, bacterial endosymbionts of fungi. Particularly, I'm going to tell you about some of our latest study that spores of arbuscular mycorrhizal fungi appear to host really highly diverse communities of endobacteria. And now I have to figure out how to switch to the next slide, so bear with me. Okay. So a bit of background on my research in general. So the main focus of my research is really on interkingdom interactions. Particularly, I'm interested in studying interactions between bacteria and fungi in the context of the plant microbiome. The plants, of course, are populated by diverse communities of bacteria and fungi, which interact with each other. And in my research, I study how these interactions affect their own individual biology, as well as that of their plant host. And so my research approach includes different scales and kind of attempts to understand these interactions at the scale of molecular biology, but also at the scale of ecology. But today I'm going to focus specifically on bacterial endosymbionts of fungi, aka or also known as endobacteria. And so the term endobacteria literally really means bacteria that live inside the cells of another organism. And in this case, that organism is a fungus. So endobacteria can populate fungal hyphae. Apologize, I'm just trying to get a handle of pointing. Okay. Endobacteria can populate fungal hyphae, and this is illustrated in this image here. It's a hypha of Rhizopus microspores, and in green, you can see it's packed full with these endobacteria. Endobacteria can be also vertically transmitted from one fungal host generation to the next in spores, and here are 2 fungal spores, also packed with these endobacteria, which now appear in white. Now, besides being just really, really super cool to study from the point of view of symbiosis research, endobacteria are actually common in fungi. And they might even be more common than we have originally thought when we started working on bacterial endosymbionts of fungi. There was this relatively recent study from Robinson and colleagues who did this large-scale screen of hundreds of bacterial or fungal cultures, fungal isolates, to see if they're associated with bacteria. And so they looked at culture collection isolates, and they also looked at existing fungal genome sequencing projects, and they found that 88% of the fungal isolates they looked at were associated with some kind of bacteria. And these fungal isolates were spread across the entire kind of all the major fungal phyla, including the Ascomycota, the Basidiomycota, but also the lower fungal, kind of lower divergent fungal phyla, the Zupagomycota, Mucormycota, Chytridiomycota, and Blastocladiomycota. This study also then did some microscopy confirmation to show that these bacteria were in fact endohyphal, at least in the ones that they looked at. So basically, it appears that bacteria associated with fungi are the rule rather than the exception. But our knowledge of these interactions still remains in its infancy. We're really just starting to understand how these interactions, what governs these interactions at all kinds of levels, molecular biology, how prevalent they are at the ecological level. And so to be able to study fungal bacterial symbioses, we need a really good model system. And because model systems are these really powerful tools for deciphering how something works at a very basic biology level. And in the realm of bacterial fungal interactions, the Rhizopus mycedohabitans system has emerged as a very effective laboratory model for deciphering these bacterial fungal symbiotic interactions. The reason it's such an effective model is because it's very, very amiable to manipulation. The fungus is easily culturable. The bacteria are culturable. You can take the symbiosis apart in the lab. You can cure the fungus from its endobacteria, put the endobacteria back. The bacteria is, there's a genetic transformation system available for the bacteria, and more recently, a genetic transformation system was developed for the fungus. So it's very, very powerful system for studying these interactions. And so the reason I'm bringing this up is because working with this Rhizopus mycetohabitans system during my time at Cornell is how I became obsessed or interested in fungal bacterial interactions in general. So I wanted to introduce it a little bit to make you appreciate it just as much as I do. And I know some of you are intimately familiar with it, so I apologize if this is a lot of repetition. But the fungus is called Rhizopus microsporus. In European high school systems, it's kind of, in all of these textbooks, it's called this they call it the common bread mold, and they study its life cycle. But really, it exists typically as a soil-dwelling saprotroph, but it can cause opportunistic infections in plants as well as animals, including humans. Other isolates of Rhizopus microsporus are actually used commercially for soy fermentation in Asia. They produce tempeh and something called sufu as well, which I'm not very familiar with, but apparently it's good. And other isolates can be food spoilage agents. These are really cool fungi just in their own right. But Rhizopus microspores can host these bacterial endosymbionts, and they're called mycetohabitans rhizoxanicum. They are beta proteobacteria, closely related to Burkholderia. And they produce this toxin called rhizoxin, which the fungus can use to be pathogenic on rice seedlings. It can cause a disease called rice seedling blight. The bacterial produced toxin is the host virulence factor. But this toxin can also protect the fungus from predation by soil micropredators such as amoebae and nematodes. And I did fail to mention in here that this bacterium lives inside the fungus, but I hope that was quite clear. During my time in Teresa's lab, I used this system to try to understand the molecular underpinnings of fungal bacterial symbioses. And so I used this combination of transcriptional profiling via RNA-seq, qPCR, comparative genomics, lipid profile analysis, and these cool observations with chemical inhibitors to show three main things. And I say I, but of course I mean we, because I had lots of wonderful collaborators. So firstly, we found that endobacteria affected fungal lipid metabolism, and they affected it in a very specific way, depending on whether the interaction was mutualistic or antagonistic. Secondly, endobacteria affected fungal reactive oxygen species metabolism, again, which differed depending on whether the bacteria were perceived as mutualistic or antagonistic. And this was really cool because it kind of indicated, it hinted at this fungal, some kind of a fungal innate immunity response, which we know very little about in fungi. We know very little about fungal immunity, so that was very cool. And lastly, we found that fungal sexual reproduction was almost under the complete control of these bacteria. Now, other lab groups working on the system have additionally shown that the endobacteria also controlled fungal asexual reproduction. So if you took the bacteria out, the fungus was no longer able to reproduce asexually. And these mold-type fungi, asexual reproduction is the main way by which they transmit to new environments. So it's a very important fitness effect on the fungus. Other studies have shown that different various bacterial effectors, as well as bacterial secretion systems, were involved in the ability of the bacteria to successfully establish a symbiosis with the fungus. And then lastly, I think I mentioned this already, that the toxin that the bacteria produce is able to protect its fungal host from predators. The bottom line that I'm trying to convey is that endobacteria can play critical roles in fungal biology, and that studying them is important for not just deciphering how bacteria and fungi interact in general, but can be powerful tools in understanding novel aspects of fungal biology in general. So for the rest of my talk, I'm going to talk about a very different fungal endobacterial system that is much less amiable to manipulation, but involves this group of globally important fungi. So this group of fungi is not only globally distributed, but they have important effects on plant health, terrestrial ecosystem functioning, and they are of rising interest in sustainable agriculture to support crop yields for growing food demands. Of course, I'm talking about arbuscular mycorrhizal fungi, which was in the talk, so I guess the title of the talk. So arbuscular mycorrhizal fungi, I'm going to introduce them a little bit if you're not as intimately familiar with them as I might be. So arbuscular mycorrhizal fungi, they are these ancient plant root symbionts, which all belong to the Glomeromycotina subphylum of fungi. So what they do is they grow into the plant root, they penetrate the plant cells, and then they grow out into the surrounding soil, and then they help the plant forage for nutrients, for essential mineral nutrients. And then they exchange those nutrients for photosynthesis-derived carbon. And up to 20% of photosynthesis-derived carbon can be allocated to AMF in this way. This symbiosis is incredibly widespread, and it's estimated that plants with AMF make up around 70% of global plant biomass, arguably making this one of the most important symbiotic interactions on our planet, with overarching impacts in terrestrial ecosystem functioning and sustainable agriculture, as they are of interest as these sustainable alternatives to non-renewable and energy intensive to produce mineral fertilizer. So what is it that I mean by that? So phosphorus in the form of phosphate is the main nutrient that AMF supply to their plant host. And in conventional agricultural practices, mineral fertilizer containing phosphate is added to the soil where the plants are grown to boost crop yield and crop growth. However, phosphate is a non-renewable resource. It is mined, it comes from phosphate rock. And depending on which estimate you look at, there are various estimates ranging from being very conservative to being very unconservative. But it's estimated that by around 2050, the demand for phosphate fertilizer is going to start exceeding the potential supply. So in light of this, AMF are of rising interest in sustainable agriculture as these alternatives to the mineral fertilizer. However, AMF provide plants much more than just, they provide plants with other benefits as well as these mineral nutrients. They are known to improve tolerance of their plant host to abiotic and biotic stresses. So for example, it is well characterized that AMF will alleviate the effects of drought on many different kinds of plants, including crop plants. And this is just an image to illustrate this. This is a corn field, and these are mycorrhizal trials. They're looking at plots that are mycorrhizal and non-mycorrhizal. And so the mycorrhizal plots, as you can see, are growing much better under this drought stress. There's also many examples of AMF being associated with increased resistance to various different plant pathogens. So they're also of interest as these, not just as biostimulants or biofertilizers, but also as biopesticides. Now, applications of AMF in agriculture seem relatively simple in theory. You know, just add some AMF to your field and reap the benefits of improved plant growth and improved crop yields. However, it's proving a lot more complex than that. And the major hurdle appears to be that field AMF inoculations don't always provide a consistent benefit to crop plants. So for example, there was this recent large-scale study where they conducted AMF inoculations across 54 fields, corn fields in Switzerland, and they looked at this parameter called mycorrhizal growth response, basically the plant growth response to mycorrhizal inoculation. And they found that the mycorrhizal growth response could range from +40 to -12. meaning that in some fields, mycorrhizal inoculations were associated with up to 40% increase in plant growth, whereas in other fields, it was the opposite. Mycorrhizal inoculations were associated with decreased plant growth. So up to 12% decreased growth. And so in this study, they tried to explain what parameters explain this range of mycorrhizal growth responses of these plants. And they showed that it was actually soil microbial parameters that explained one of the major difference in the plant growth response to AMF, kind of indicating that interactions with microorganisms likely play a very vital role in the plant AMF symbiosis. So with that in mind, bacteria are emerging as these important new players in the plant AMF symbiosis. For example, it turns out that AMF have what's called a hyphosphere microbiome, meaning that the extra-radical hyphae of AMF are populated on the surface by specific sets of bacterial taxa. And this hyphosphere microbiome can have important functional effects on the plant AMF interaction. For example, there is experimental evidence showing that the hyphosphere microbiome improves organic phosphorus mineralization, meaning that the hyphosphere microbiome can help the AMF provide these mineral nutrients to the plant host. There's also this microbiome, hyphosphere microbiome also has some other hypothesized functions, which are, I think, being tested at the moment. So it's hypothesized that it can improve organic nitrogen mobilization. It's also hypothesized that the microbiome can protect the AMF host from bacterial predators, and even probably even providing some alternative carbon sources to the AMF host. So the studies on the AMF hyphosphere microbiome mainly focused on the extra-radical hyphae of AMF, so the hyphae that are in the soil. And we've recently published some interesting results which indicate that there may be a similar recruitment of specific bacterial taxa onto the AMF hyphae that are actually inside the plant root. So in this study, we did some multivariate modeling to show that there were significant correlations between the AMF and bacterial communities that coexisted together inside plant roots. So there's potentially another hyphosphere microbiome that's inside the plant roots. Now, besides interacting with bacteria that are associated on the surfaces of their hyphae, AMF host endobacteria as well. So they can host these endobacteria inside their hyphae and inside their spores. And to date, 2 such endobacteria have been studied and described and named as new bacterial species. So one of them is called Candidatus glomeribacter jagospirarum. Candidatus status means you can't culture them. I'm just going to call it glomeribacter for short. Glomeribacter is the better studied endosymbiont in terms of functional characterization for what it does for its fungal host. It appears to possess characteristics of mutualists in that it primes the energy metabolism of AMF. It can increase, so glomeribacter is associated with increased hyphal proliferation of AMF during spore germination and also increased sporulation success of AMF. The other endobacteria are called Candidatus moyiniplasma glomeromycotorum. Teresa named this awesome new species. My colleague tells me that there is a kind of cocktail called Moini. So he has dubbed it Moini, and I like it. But I'm going to call it moyiniplasma today. So I think it sounds wonderful. So moyiniplasma is different from glomeribacter phylogenetically, and it's also different in that there is no, we still have no idea what it actually does in the fungus. There is no experimental characterization of their role inside AMF. But there are some molecular evolutionary studies that indicate that moyiniplasma endosymbionts, they possess characteristics of parasites. And so when I began studying the AMF endobacteria symbiosis, everything we knew about this relationship came from AMF derived from culture collections. and nothing was really known about them in nature. We didn't even really know, we thought they should be, but we didn't know how often they were found there and if they're there in the first place. So during my time at Cornell, I also began looking at the distribution of endobacteria in these natural AMF populations. So to do this, we went out to a natural dune ecosystem located at Cape Cod. Whenever I mention this, everybody laughs because they think it was just great because it's a wonderful holiday destination. But we went in November and it was quite miserable. So we went out to Cape Cod. We took soil samples. From these soil samples, we extracted AMF spores. We surface sterilized them, and then we screened them using PCR for the presence of the two known endosymbionts, Mycoplasma and Glomeribacter, using specific primers. We found that Glomeribacter was actually very rare in the AMF, found only in around 2% of the spores, whereas Mycoplasma were very, very common, found in about 88% of the spores. We also detected potential presence of a third type of previously uncharacterized endosymbiont in these spores that were potentially related to Burkholderia. And this sort of came about as a result of our Glomeribacter specific primers not being particularly specific and amplifying other Burkholderia species. So this can be seen from this tree. On this tree, the sequences highlighted in red and blue are sequences that we amplified from the spores. And you can see that some of them group within the known Glomeribacter clade, whereas other ones instead group with free-living Burkholderium. And so we concluded that it's possible that a third type, novel type, a previously uncharacterized type of endosymbiont could live inside these AMF spores. So after my PhD, I moved back to Ireland, and I wanted to continue characterizing the distribution of these endobacteria in natural AMF populations. So we went out and we sampled the two different sampling sites in Ireland. One was, again, this natural dune ecosystem site, and another one was a nearby agricultural grassland. Both of these were located on the east coast of Ireland. And at first, we did the same thing. We extracted spores directly from the soil. We surface sterilized them, and then we screened them for Glomeribacter and Mycoplasma. We found that just like at Cape Cod, Glomeribacter, which is considered to be a mutualist, was very, very rare, only found in about 1% of the spores. Whereas Mycoplasma, the one that we don't really know what it does, but maybe it's a parasite, was very, very common, found in around 80% of the spores. And now this is not an average of these two sites. This pattern was the same, almost identical for both of those sites. And it was, as you remember from my previous slide, are almost the same at Cape Cod. So this pattern of distribution must mean something. We just don't know what it means yet. But I then decided to characterize the entire diversity of endobacteria associated with these AMF spores. So we took all of the AMF spores that tested positive for presence of any type of endobacteria, and we subjected them to total bacterial community profiling using 16S amplicon sequencing on this Illumina MySeq platform. And in this way, we characterized 84 AMF spores. And to my great surprise, the diversity of endobacteria that we discovered was very, very, very high. So here you're looking at relative abundance plots of bacterial phyla. This plot is showing presence of these phyla in spores kind of split by the sampling site in the agricultural dune and the agricultural site and then the dunes. And this plot here shows the same thing, but each row corresponds to a single spore sample. So firstly, you can see the plots appear red. In red are the bacteria from the Tenericutis phylum. I think they've recently been renamed. Now they're called Mycoplasmatota. But these are the Mycoplasma endobacteria. So you can see that the Mycoplasma endobacteria are the most commonly found, but also have the highest relative abundance within individual spores. Now, Glomeribacter belongs to this Proteobacteria phylum, but the Glomeribacter sequences, the Glomeribacter was so rare that the Proteobacteria that you see here were not Glomeribacter, they're just other endobacteria. But the point is that from these plots, you can see that we detected endobacteria in these associated with these spores belonging to 10 other bacterial phylum. And in fact, an individual AMF spore could be home to as many as 277 other bacterial OTUs, which is an approximation for bacterial species, that were not Glomeribacter or Moeniplasma, with an average of about 25 other bacterial OTUs detected per spore. Now, these bacteria are mainly composed of known free-living and soil aquatic bacteria, and they're not previously known to have any kind of an endofungal lifestyle. So this sort of challenges and changes our view of AMF being able to host two different kinds of endobacteria. And in fact, it turns out that AMF spores can be host to whole communities of endobacteria. But what the heck are they doing there? Well, there was some really interesting work published recently by Nanditha Venkatesh and colleagues from Nancy Keller's lab, where they showed that in other fungi, free-living bacteria can invade these fungal chlamydospores, and chlamydospores are these environmentally resistant types of fungal spores that certain fungi produce. So these free-living bacteria could invade the chlamydospores of these fungi, and then being inside the chlamydospores made them more resistant to environmental stresses, such as low temperature stress or low nutrient stress. So in this study, they called these bacteria hitchhikers, which hitchhiked, inside fungal spores. And they sort of they hypothesized that being able to utilize these chlamydospores could explain the persistence of certain bacterial taxa under harsh environmental conditions. And so this led us to hypothesize that perhaps these diverse communities of endobacteria that we found in AMF spores could analogously be these bacterial hitchhikers. And it's not so hard to believe. AMF spores are packed full with nutritious and delicious lipids and glycogen, which could be used as a food source for these bacteria. And they could potentially shield the bacteria inside their spores from other environmental stresses. So I then took a closer look at the Moeniplasma population structure inside these individual AMF spores. Now, Moeniplasma endobacteria are an unusual type of endosymbiont in that they can be incredibly diverse, even within a single AMF spore, with up to 3 Moeniplasma OTUs having been previously known to coexist inside an individual AMF spore. Incidentally, Glomeribacter endobacteria endosymbionts are always genetically uniform within AMF isolates. Now, in our work, in our field, AMF spores, we found that a single AMF spore could be host to up to 50 Moeniplasma OTUs, or species, with an average of about 22 OTUs found in an individual spore, kind of highlighting this previously unknown diversity of even these endosymbionts in environmental AMF isolates. Now, such high intra-host diversity of endosymbionts is actually considered to be a hallmark of parasites because this diversity is expected to increase competition dynamics among the symbionts at a detriment to the host. And so, in fact, this high intra-host diversity of Moeniplasma populations is why they were hypothesized to be parasites in the first place. However, Moeniplasma are very, very common in field AMF isolates, with about 80% of all of the spores that I've screened so far harboring Moeniplasma. So I think it's very unlikely that Moeniplasma would be maintained to such high levels in AMF populations if they don't provide any benefit to their host, because it's expected that selection would remove such parasites from populations. So I'm instead proposing that it's possible that Moeniplasma might actually act as conditional mutualists of AMF. And in a conditional mutualism, the symbionts provide a benefit to their host under specific environmental conditions. And in this realm of bacterial fungal interactions, there's evidence that endobacteria can provide a conditional benefit to their fungal host in the presence of predators. So I've already talked about the Rhizopus mycetohabitans system at the beginning. So the Mycetia habitans endobacteria produce a toxin that protects its fungal host from soil micropredators. And analogously, in the symbiotic system between Mortierella and Mycoavidus, the Mycoavidus endobacteria produce a toxin that can protect its Mortierella fungal host from predatory nematodes as well. So in this case, the endobacteria are acting as defensive mutualists of their fungal host. So analogously, we're proposing that Moenoplasma endobacteria might act as defensive mutualists of AMF. And this hypothesis sort of came about through our observation that spores of AMF that harbored higher diversity of Moenoplasma harbored less diverse communities of all of these other potentially hitchhiker endobacteria. And so you can see it on this plot. On the x-axis, I've plotted the Moenoplasma diversity, and on the y-axis is this diversity of all these other endobacteria. And each dot represents an AMF spore. So you can see that spores that harbor higher diversity of Moenoplasma harbor lower diversity of all these other endobacteria. So I'm thinking, or we're proposing, that by maintaining this high population diversity within AMF, Moenoplasma are likely more effective at outcompeting all of these other hitchhiker, potentially detrimental, potentially parasitic endobacteria. Of course, this requires experimental testing, and this is something that I hope to do in the future. And lastly, looking at the Moenoplasma study, like looking kind of closely at these Moenoplasma populations, we found that Moenoplasma population structure, Moenoplasma richness, Moenoplasma incidence rates across the spores, and also the Moenoplasma intraspore relative abundance were all related to host genotype, meaning that all of these measures varied according to the AMF host genotype. So for example, in this PCOA plot, which is plotting the Moenoplasma populations and how close and how similar they are. So the closer the dots are, the more similar those populations are. And it's colored by the AMF host genotype. So for example, in green, you're looking at Moenoplasma populations from diverse spores. And this grouping according to AMF genotype is significant. Now, it's also significantly different across the different sites. So the agricultural site or the dune site. But you can see at the bottom here, we're looking at the Moenoplasma populations from Scutelospora AMF. And you can see that they grouped together, despite the fact that they came from two very different sites. One was an agricultural site, one was a dune site. So it is really the host genotype that seems to be driving this population structure. So leading us to conclude that Moenoplasma populations are likely, Moenoplasma population assembly is likely driven by host genotype. And this is sort of analogous to what has already been observed and shown to be true in other more popular eukaryotic systems, such as humans and mice, where host genotype is a known main driver of microbiome community assembly and community structure. So in humans, in mice, but also in plants. And more recently, this is also shown to be true for fungal fruiting bodies. And so sort of we're now showing that this also extends to AMF and their endobacteria. So currently, as Teresa said, I'm quite a new PI and I'm in the process of setting up my lab. I have named it the MINT lab. For some reason, I just didn't think Lastovetsky lab had the same cool ring to it. So MINT lab stands for microbial interactions. And a friend of mine did this really nice logo. But one of the current main research directions in my lab is that we're trying to functionally characterize Moenoplasma, trying to finally figure out what it is that they do in AMF. And the reason this hasn't been done before is, as I mentioned at the beginning, AMF are not very easy to culture. They don't like being cultured. They actually hate it. They're, of course, they're obligate biotrophs, so they absolutely require the plant host in order to be able to complete their life cycle. So it's been hard to try to obtain these isolates that are genetically identical in every other sense, except differ in the presence of these endobacteria. So I have two very hardworking PhD students with whom at the moment we're trying to generate these isolates. And once they have them, we can compare them and address the role of Moenoplasma in AMF biology, but also in the interaction between AMF and the plant host. Another thing that I would like to do is try to untangle these hitchhiker bacterial communities. How are they assembled? Where do they come from? Do they come from the soil? Do they come from inside the plant root? Do they have any mycorrhizal helper function? And where are they inside these spores? We know they're not on the surfaces because we're surface sterilizing them, but where are they inside the spores? There's this really cool images that were, in this quite old publication where they were trying to validate a spore surface sterilization method. And as you can see, there is no bacteria or anything on the surface, but they found these bacterial-like cells or structures and embedded within the spore. And they also found these even small cell-like structures, which in size are smaller than any known bacteria. So that's what I would love to find out. Where are these different communities of the endobacteria in relation to the fungal spore in the different compartments? And where do they go when the spore germinates? Do they continue living in the hyphae? Do they kind of jump out and live in the soil or somehow associate with a plant? Those are really all cool questions, I think. And another direction that I want to pursue is looking, again, kind of going back to my PhD roots and looking at these kind of earlier diverging Mucormycotina fungi and also Mortierella mycotina from soil and from food. And this is just an image of this mucor isolate that we isolated over the summer that has a very potentially has a very cool type of symbiont. And lastly, I just wanted to highlight for those of you that are thinking, that are graduating in the next couple of years and are thinking of going to Europe for a postdoc, there are these cool postdoctoral fellowships that the European Union gives out to the worthy students. They're called the Marie Curie postdoctoral actions. They are very well funded. You get a very nice stipend from them. So they're very good if they're mobility fellowships, so you have to go to Europe to study, or if you're going within Europe. So that's just to highlight, that's a really good opportunity in general. If you're interested in working with me, I'd be happy to talk to you about it. The current call is closed, but it will open again the next 6 months. So it'll be, deadline will be again next September, we think. So with that, my acknowledgements. Thank you very much, first of all, for all of you for listening. Thank you again for the invitation. Thank you to my wonderful collaborators, to my 2 PhD students who are hard at work in the lab at the moment, and for the Irish Research Council for the funding. And with that, I'd be happy to take any questions.
Could you tell us more about how your students are trying to get the cultures without the endobacteria? What are some things you can try to get that question?
Oh, so thank you. So I have a mic. So the question is, how is it that we're actually trying to get these genetically identical isolates that do and don't have endobacteria? We have reason to believe that once you have an isolate that has MRE in it, that we're going to try to serially propagate it for multiple generations and get an endobacteria-free isolate. This is how, in the studies of Glomeribacter, they're able to cure the AMF fungus from Glomeribacter. The alternative, of course, is how we do it with Rhizopus, is using antibiotics, but this has proven to be unsuccessful in the past. But who knows, this might be something we'll try as well. Mm-hmm. Yes, Esther.
Have you looked at all, that last EM really intrigued me with those tiny.
Mm-hmm.
Have you looked at all for viral sequences and anything, genome assemblies or anything?
That's a very good question. The question is, have I looked at any viral sequences in mycorrhizal assemblies? I haven't, but other people have, and AMF are known to have mycoviruses. Some of them, I think some taxa are more prone to have them, but it's very, as far as I know, Teresa might be able to comment more. As far as I know, it's nobody's doing this just yet, at least. Yeah, it'd be cool to do.
It was intriguing because it almost. looked
Those
like a
small,
little
yes, those small cells. So they are, as far as I remember, they are about 0.1 microns in size. All right, here, these small cells. And mycoplasma, remind me, are just about over that. So it could be, I mean, some bacteria are known to kind of shrink, right, when they become dormant. Could be mycoplasma shrinking as it's being dormant, but that's completely, I'm just making this up. So yeah, it could be viruses or kind of on the bigger side, or it could be small bacterial dormant cells.
I may have missed this, but is the population of bacteria in the AMF spores the same as in the hyphae?
So the question is the population of endobacteria in the spores the same as in the hyphae? And the answer is we don't know. In terms of the known endosymbionts, they are known to be in the hyphae. But the newly discovered symbionts, we don't know. And that's something that I would like to do. Yeah. Adam.
You know, it was a great talk. I hate to do this, but I have to play the role of reviewer 3 and ask you, how are you certain that the taxa you're detecting are inside and not somehow protected on the outside? In a biofilm or through some mucilage that's on the spore, protected from your sterilization technique?
We haven't done any microscopy to confirm this, but the method that we're in, we don't do this every time, but this method is well established for being able to clean everything that's on the outside. It's a combination of hydrogen peroxide, chloramine T, and tween, and lots of different shaking and everything. And this study where I've pulled this image from, they were doing just that. They were trying to figure out why were they still having sometimes bacterial contamination from their AMF spores when it was clear that their sterilization technique, they did microscopy, removed everything from the outside? Yes.
In your graduate work, when you were looking, you were sort of characterizing how much glomeribacter and mycoplasma were in different sites. When you saw, I think it was glomeribacter was 2%. When that signal came up, was it like a high amount of signal, or did you think then that it was sort of like lower levels? Maybe it was in other things as well, but it was at lower levels and indetectable. Or was it got some spores and took them over versus mycoplasma and had most of the spores and took them over?
So I think you're asking about how, in terms of relative abundance inside individual spores, how much glomeribacter is there when it is there?
Yes.
So when they were first study in Cape Cod, we didn't do that. We just did PCR, so it was just plus minus, the presence absence. In this study, I did have a look at this, and glomeribacter were always relatively rare. And actually, I have this whole section in this paper where I describe how I was trying to actually find them. I had to really dig to pull out glomeribacter sequences and separate them from all of these other sequences, because using conventional, microbiome analyses, glomeribacter were not coming up at all. So I had to kind of spike the database with known glomeribacter, known related, Myceta habitant sequences to be able to pull out anything that was glomeribacter. And then I had to do some other things to find, there was really just 3 or 4 glomeribacter OTUs across all of the spores, and they were always quite low in relative abundance inside individual spores. I can't give you a number because I can't remember, but happy to send you the data if you want to have a look. Mm-hmm.
I have a question. So, in relatives of mycoplasma are found in Montero mycotina. Are they also present in other fungi? And we just don't know about this because we have not looked.
Mm-hmm.
And what would be their functions? What would you speculate about their function in Montero mycotina first, and they're present in other?
Are you talking about the mycoplasma related endobacteria in Mortierella, and what do I think is their speculative function? So I'll answer the question first, and whether they're present in other fungi. So when I was characterizing the mycoplasma populations in AMF spores, I did, like I said, I spiked the database, the database which I was using to pull out the mycoplasma sequences. And some of them were more, I didn't do any phylogeny because these are really short reads and you can't do phylogeny with them. But some of them did group closer to the reads from endogeny, for example. And some of them more closely related to Muniplasma from, actually, I don't want to lie, I can't remember this, but definitely remember the endogeny. I would imagine some of them, yeah, so I think the whole diversity of Muniplasma is still quite, we still haven't characterized it entirely. And it's entirely possible that there are some more related sequences to Mortierella of Muniplasma living in AMF. As to their function in Mortierella, I can't comment on that entirely. But like Teresa said, they are very closely related endosymbionts that living in another type of fungus called Mortierella mycotina. And they did some functional, they were able to cure some Mortierella isolates from these Moeniplasma, mycoplasma-related endobacteria. And they showed that under different laboratory conditions, the Moeniplasma-related bacteria could be detrimental. They reduced fungal growth. So this was kind of another nail in the coffin of saying, oh, they're pathogens. But it's entirely possible that they do something, in a conditional mutualism sense, that they might do something under certain environmental conditions that are positive for Mortierella actually out in the real world.
This has been a production of Cornell University, on the web at cornell.edu.